Kia ora, hello everybody. My name is Renee Johansson and I work for the Beyond Myrtle Rust program. Thank you for joining us for our monthly webinar series, bringing you the latest in Myrtle Rust research. Today's speaker is Roanne Sutherland, post-border biosecurity researcher at Scion. Roanne is well known to many of us involved with Myrtle Rust research in New Zealand, as she has been active eyes on the ground for several years now, both as part of her job and as a concerned citizen. Her most recent notable find is of the disease on Great Barrier Island, from where she lodged a report on iNaturalist just last week. Roanne, you can commence your presentation.
Yeah, thank you, Renee, for the introduction. And I'm really excited to share some of the research that Scien's been doing over the past 3 years in the ngahere, in the native forest. So hello to everybody online. I'm just going to start with a quick overview of Myrtle Rust in New Zealand for those of you online that might be new to the webinar series. Myrtle Rust, Austropuccinia psidii, was first detected on Raoul Island on Kermadec, pōhutukawa in March 2017. And 2 months later, it was found in the mainland on Kerikeri. Soon after that, it was found across the North Island in the Bay of Plenty, Waikato, Taranaki, and East Cape areas, and is now established across all regions in the North Island and parts of the South Island, including the latest find in Christchurch and on Aotearoa, Great Barrier Island, as Renee's already mentioned. Now, Myrtle Rust is an airborne biotrophic pathogen that requires a living host to survive and reproduce on. Myrtle Rust only attacks species in the Myrtaceae family, and in New Zealand we have 30 natives, and these include pōhutukawa, mānuka, rātā, mairitawaki, and many introduced species such as wheejoa, lily-pilly, and eucalyptus. The pathogen only attacks new growth, which include leaves, stem, flowers, and fruit. It does not attack the woody trunk or roots. Myrtle rust can reproduce both clonally and sexually, and hosts have a varying susceptibility to the pathogen, and there is also varying susceptibility within populations. So one of our most vulnerable natives at this stage appears to be Lophomyrtus species. This entire genus is endemic to New Zealand and consists of 2 species, ramarama, Lophomyrtus ballata, and Rōhutu, Lophomyrtus obcordata. With the arrival of Myrtle Rust, the conservation status was raised to nationally critical in 2018. Both are evergreen shrubs to small trees, although trees have been recorded to reach heights over 10 metres. Both have patchy distribution across the North Island and South Island, with flowering occurring November to March and then producing small edible red berries. So a little bit about ramarama. Ramarama have distinctive bright green oval bubbled leaves and edible berries that were part of the traditional diet for Māori. Rōhutu has small, thick, heart-shaped leaves, and plant parts were traditionally used in rongoa practices. These species naturally hybridize, creating a fully fertile hybrid with a range of leaf shapes and sizes. Nursery cultivars like Red Dragon are commonly planted in urban gardens across New Zealand. In 2018, 2 sites were chosen in the Rotorua district on the edge of remnant forest, approximately 13 kilometres apart, where Lophomyrtus and other Myrtaceae species are present growing as natural populations. Lophomyrtus trees along the transect were selected for their accessibility for visual checks, and monitoring started in October 2018. Each tree was assessed fortnightly, along with other Myrtaceae species within a 3-metre radius. There was an abundance of seedlings under mature trees, and 2 seedling plots were set up at each site. Site A was our infected site, where Myrtle Rust was first detected in 2018 on 2 Lophomyrtus trees by Jackie Bond at Dock. Site B was Myrtle Rust free when monitoring started and was monitored monthly until the pathogen was detected, and then fortnightly monitoring commenced. So along those transects, we chose 5 Lophomyrtus trees, and then we had additional species as well that was outside of these transects, which include mānuka and pōhutukawa, and then rātā species as well, Myrtaceae diffusa, Myrtaceae fulgens, and Myrtaceae perforata. We also have other monitored sites across the North Island in the Kaimai-Mamaku Ranges, Auckland and Taranaki, and these were established in 2020 and are monitored monthly over summer. To assess the disease severity, we used the New Zealand Myrtle Rust monitoring form, which was developed through consultation and testing with many organisations across New Zealand, and was completed in June 2018. Below, there is a picture of Nick and Karen testing out the form at Plant and Food Campus in Auckland. So the monitoring form includes 4 sections: site description, plant description, environmental disturbances, and disease severity. The site description and plant description only needs to be completed once. Then we record fortnightly records of environmental disturbances and disease severity. Data on plant phenology and canopy density are also recorded. All parts of seedlings and smaller trees are inspected, but for larger trees, only a percentage of the tree foliage is able to be assessed. In these cases, canopy coverage was recorded to determine changes in density over time. So what were some of the impacts that we saw on Lophomyrtus? So Myrtle Rust is most active in New Zealand during the warmer weather. The Myrtle Rust season is considered to be from around October through to May. At Site A, in the first season, 2018 to 2019, 60% of the trees had Myrtle Rust symptoms, with 80 to 100% of the new flush infected, which led to defoliation and dieback. Over the second season, 2019 to 2020, 80% of the trees were infected, and after 3 seasons, all of the Lophomyrtus trees were infected. So at Site B, you remember this was our uninfected site to begin with. Over the first season, no Myrtle Rust symptoms were detected. During the second season, Myrtle Rust was seen for the first time on 40% of the trees. In the third season, all Lophomyrtus trees became infected. Defoliation of new growth occurring within 3 months after the first signs of infections, which you can see in the photos below. In December, the new growth was covered with Uridinio spores, and by March, all of those infected leaves had dropped off. So the 2 photos on the left-hand side show the extent of defoliation after 3 seasons of reinfection, with only older leaves remaining. We currently don't know the lifespan of mature leaves, and this will be a factor in the survival of these trees, as each season, all new flush is killed. So tree death is yet to occur at these sites. However, death of mature trees has been recorded on the East Cape after 2 to 3 years of reinfection. So localised extinction of Lophomyrtus is probable. So the seedling plot monitoring. So Lophomyrtus seedling plots were established at both sites in 2018. At Site A, the infected site, 98% of the seedlings became infected in year 1, with 20% dying after 2 seasons. And in the one plot, there's been no new seedling recruitment has been recorded after the first season of infection. So here's some pictures of the seedling plots. It's a little bit hard to see, but hopefully you can make it out. The top 2 photos show one of the seedling plots, plot 2 at Site A, where all the seedlings have been impacted after 3 seasons and no seedlings have survived. So the lower 2 photos, this is at Site B, plot 1, and shows the seedling growth and recruitment that's happened over 2 seasons when they were Myrtle Rust free. This last season, they started to get infected, although we have not seen any seedling loss occur yet. So some of the other hosts that we have recorded Myrtle Rust symptoms on are 3 climbing rātā, Myrtaceae perforata, Myrtaceae diffusa, and Myrtaceae fulgens. We've also seen infection on mānuka seedling, and at sites across New Zealand, we've seen infection on mighty tāwaki. So the importance of long-term monitoring. So these are the first long-term monitoring sites to be established in New Zealand and have provided critical information on impacts along with valuable observations. Continued monitoring is important as it can show how the pathogen continues to spread within sites into new areas and onto other host species. Long-term monitoring not only records disease impact and decline, but it can identify ecosystems at risk, seedling recruitment, phenology data, and can identify tolerant or resistant species within a population. Field observations have recorded potential natural enemies already present in the native forest. During regular monitoring, a minute larva has been seen feeding on the rust spores over the summer. The larvae has since been reared in the lab and used morphological and molecular tools identified as a new Mycodiplosis species, and has been seen in the Bay of Plenty, Auckland and Taranaki areas. We've also seen white mycelium covering Myrtle Rust pustules on fruit. This was recorded in the field, and in the lab, it was seen to be completely invading the Myrtle Rust pustules. Culturing the mycelium, we were able to isolate 2 cladosporium species, which are known to inhibit development of other rusts and could be a potential hyperparasite of Myrtle Rust. So research on both of these is ongoing. So Lophomyrtus phenology. So all reproductive structures of Lophomyrtus are highly susceptible to the pathogen, which include flower buds, flowers, developing and mature fruits. Infants will brown and dry, as seen in the far right photo, and then they prematurely abort. Using existing transects, Lophomyrtus trees were selected that were of reproductive age and with branches that were easily assessed from the ground. Over 50% of the developing fruits became infected and subsequently aborted. Very few fruits escaped the pathogen to reach maturity. This last season, 40% of the trees at Site A did not produce any mature fruits. Continued reinfection will reduce seedling recruitment across the forest. This work is still ongoing with funding through Ngā Rākau Takitaki. So this next slide, we've got a couple of photos. So this is showing some of the infection on the inside of the fruit capsules when we open them up. And you can also see that there is spores sitting on top of the seeds. And we're yet to understand the full impact of Myrtle Rust on seedling viability. So what are some of the wider ecosystem impacts? So it's really important to understand what these might be and other biota associations that may be impacted if widespread loss of Lophomyrtus occurs. During regular monitoring, numerous invertebrates have been seen on Lophomyrtus trees feeding on infected and uninfected leaves. A review revealed that there are 109 known species that have a direct association with Lophomyrtus. And this is across 52 families, including invertebrates, vertebrates, vascular plants, lichens, and fungi. This was an initial literature review and database check, and more research is also needed in this area. So I just wanted to highlight one of the student projects that were done over summer. So this is completed by Kristen Gillard. The aim of the study was to investigate how canopy density and edge gradient Myrtle Rust. So the main findings was at both sites, as canopy density increased, there was a significant decrease in disease severity. This is some really interesting work and something that we hope to continue to look at over the coming seasons. So gearing up for summer, we've got a little bit more funding through Ngā Rākau Takitaki to look at Lophomyrtus light requirements for reproduction and important pollinators. So field observations have recorded native bees, weevils actively visiting Lophomyrtus flowers. However, there is likely to be more pollinators of these species, and it is unknown if these insects also pollinate other species or are potential vectors. This information will help improve our understanding of Lophomyrtus and its role in the wider ecosystem. So there is still more that we need to know. More information is needed on the basic biology of these species and their role in the ecosystem. A national surveillance and monitoring program is needed as we lack knowledge of the spread and severity in the ngahere. We need to know what associated species will be impacted or lost as these species declined in our forest ecosystems. Tree death has already occurred where climatic conditions is conducive to year-round infection, and where else in New Zealand is this also occurring? And there is more funding that is needed for operational science. It is critical to act now to reduce the impact on biodiversity before it's too late. So as always, I'm one person from a team of many that have been working on this research, and we've had many funding parties over the year, and I'd like to acknowledge everybody, including scientists, technicians, mana whenua, tangata whenua. And then we've got time for questions.
Thank you very much, Rowan. That was an excellent talk. Now, it's ironic that we did have a few technical difficulties there, given this is in fact a re-record due to some technical difficulties, but you know what? All the information is out there. That was really informative for people. And if you do want to stick around and watch this recording, dear viewer, you're now going to see a slight aberration, a likely mysterious change in the background of Rowan at least, because we're going to cut on our very lively and informative question and answer session from the live recording earlier in the week. Let's go to that now. Excellent, thank you very much, Rowan. It always strikes me that it's a strangely disconnected experience giving a webinar and there's no applause, so I'll give you some applause. Maybe I should make this a thing in the webinar series from now on, because, and we do have this question here, starting with the awesome talk, Rowan, well done. Very good indeed. Just wondering about the infected fruit. Now, of course, that was a very dark set of results there, unfortunately, and so I know myself, a couple of my questions hinge on where is there hope, and perhaps this person is thinking the same thing. Did 100% of fruit that were infected die, or did some make it through despite some small level of infection? How doomed are the fruits, Rowan?
Yes, so so I haven't had a chance to analyse all the data yet, but yes, there were some fruits with small amount of infection that managed to get through, but that infection only occurred when the fruit was more mature. So in those early stages of fruit development, they didn't survive if they became infected.
Right, now don't have any more questions from the audience, so audience, if you have got something on your mind, do type it in, and in the meantime I picked up on a couple of things myself. So that result about canopy density was very interesting, given what we do know about this disease in Austropuccinia psidii, and so we're saying canopy density seems to have lower levels of infection. Have you guys been, yes, but someone has typed the same thing in, considering the usual relationship between humidity and fungal infection, is this decreased infection with the canopy density unusual? Have you guys had a go at speculating about what might be going on there?
Yeah, so we have speculated, and I can take a best guess as to why it might be happening, but it is something that we definitely need to look into further and get some funding to do more research in this area. It could be microclimate conditions in there. It also could be that you've got a lot of other leaf material that the spores might accidentally attach to and can't find their host plant. There's lots of other things that could be going on there that we actually need to look into.
Yeah, well, this is the thing, isn't it? Is it correlation? Is there something else going on as the canopy gets denser, especially in terms of greater variety of species there, and less inoculum pressure, and so on, that might be driving this result? And I did wonder, therefore,
Yeah.
are you looking at plants under more dense canopy areas, it just looked like in your transits they were somewhat on the edge. Guess this is the clinging for hope, Roanne. The edge one's doomed, but the other one's not so much.
Yeah, look, definitely on the edge. They are really susceptible to the pathogen. They've got, the right light requirements and for the plant to grow, so that they're also the ones that are flowering and fruiting. So they're also the ones that are being impacted. It would be great to do some more studies in some denser forest, but yeah, we need more money.
Always the case, isn't it? All right,
Yeah,
good. Now, we
it is.
have a lot of questions, but I'm going to be selfish because I can tell we've still got 7 minutes left, and once I get to those questions, we ain't coming back to mine. And I am curious about that Mycodiplosa species. I know nothing much about invertebrates. What might this organism have been eating before Myrtle Rust got here, and what else do these things eat?
Yeah, so it is a rust feeding larvae. So
Okay.
prior to Myrtle Rust's arrival, it could have potentially been eating a native rust or another rust. This is something we're also unsure of. We've only just had an ID, and it is a new species. So potentially it could be a native species, but this is all very unknown at this stage.
All right, very good. All right, back to the audience now, and I think this too might be a little optimistic. Is it possible to stimulate Lophomyrtus to fruit or flower outside the Myrtle Rust season? Gosh, I don't know a great deal about that kind of thing. What about you, Roanne?
Um, Maybe in
In a
yeah,
greenhouse with a
actually
bunch of the
I'm
right
not sure.
hormones.
Yeah, definitely not in the
You can
native
do it.
environment.
Yeah, that's right. You can do anything you like if you've got the right hormones.
Yeah, but yeah, I
Rightly
don't think
from
you'd be doing it out there in
Plant
the native
Science
forest.
101. Yeah, you're not going out in the forest with some hormones, probably. Right.
No.
I see there is an app for the public. This is indeed true. Citizen science can assist with monitoring by putting records on iNaturalist. Have they been of much assistance so far?
Look, most of the records that come up onto iNaturalist are in the urban environment. Um, there is maybe one through citizen science that has been recorded in the native forest. I do put my records up there, but I'm not counting those in that. But yeah, mainly it's urban environment where people are recording infection, not in the native forest, and that's where we really need to understand how bad it is, where is it, what's going to be the species impact, how big are those populations that are being hit by myrtle rust.
Yeah, it's very tricky, isn't it? I mean, certainly as a research program, we're encouraging the use of iNaturalist, but with full awareness of the drawbacks of this approach. And the fact is that this approach will never replace a fully funded national surveillance / monitoring system. That the value that that would bring is not going to be matched by citizen science, but rather ideally the two would work together. And we
Yeah.
certainly wouldn't want to discourage anybody ever from putting a record up there. It had been revealing some patterns, and certainly some new knowledge has come through that mechanism. All right, the next question, is it photosynthetic sugar content of plants? I wonder if the questioner means these particular species are being hit. Is it something about the leaf chemistry and the sugar in the leaf? Maybe, not
Yeah, I
sure.
don't think that that's known. It's
I
just
doubt it.
yeah,
Isn't it? I
it's
doubt it's
just that
known.
new susceptible growth that's soft. It's probably easily penetrated by the myrtle rust. Yeah,
Yes, of course. I mean, perhaps if this question is about why it's getting the new growth, that is a very good and very interesting and very technical question, which is something that next month's webinar speaker may be able to address for you. Telling you about his talk a little bit later on. Can we please have access to this webinar? Yes, certainly. All of the webinars go on the Beyond Myrtle Rust website. Anybody can view them there, and anybody who registers will also have a recording emailed. Usually I do it a couple of days afterwards. We had some technical difficulties actually with the last webinar recording, so it was about a week before I managed to get that out, but hopefully we don't have any technical disasters today. All right, is there any research looking at seed conservation and resistance for ramarama? Another very good question. Not in the
um,
Beyond Myrtle Rust program. What do you know about Roanne?
Yeah, I mean, so I guess that there has been a lot of seed collection done by DOC, and also done by mana whenua, where they are storing those seeds. And Curran down in Wellington is looking at seed storage of some of these soft fleshy fruits, like
Yes,
ramarama
yes.
and Rōhutu and Mairi Tāwaki.
Yes,
What was
yes,
the
she
second part
certainly
of that
is.
question?
Resistance for ramarama. So, well, we were just talking before about work going on at Auckland Botanic Gardens in terms of looking at resistance for garden cultivars, and some has been found.
Yeah, look, and that's a really interesting question. And I think that there is the potential to be able to see resistant species within a population. They've actually found this over in Australia, where they've gone back to areas and all the trees are dead except for one that's
Wow.
obviously somehow found some kind of way to overcome the pathogen. So there is a potential we might see the same thing here, but we need to be out there looking for it.
Absolutely. This just brings us back to that same point, isn't it? This kind of knowledge could be out there ripe for the picking, and we're missing it right now. Right. Interesting question. Referring to your slide that you've got there, what are the white things on the underside of the leaf middle picture second row? They stood out to me as well.
They are moth eggs.
Oh, so there you go. Simple question, simple answer. Right. Well, yes, this is a crucial problem, says this person. For New Zealand ecosystems, an issue we should all be aware of. Ha, well. What advice is there for people involved in forest restoration projects? Do we not plant Rōhutu or ramarama? I have my own thoughts on that, but I better stop feeling tempted to take over the question and answer session. And perhaps, do you have thoughts, Roanne? Is that something you're prepared to discuss in this forum?
Oh, look, I think that's a really tricky question. We don't want to just stop planting our endemic species, but at the same time, you know, if you're planting highly susceptible species, it can build up inoculum load in that environment, and that can then impact on your other natives that are
Yes.
somewhat more tolerant to the disease. So I've talked a little bit about the climbing rātā. We've only seen infection on the climbing rātā when it's really close to infected Lophomyrtus. So without the presence of Lophomyrtus, would those species have been infected? Not quite sure. So I think
Yep,
that's
that
going
is
to
a
be
very good point.
up to the individual moving forward. But I would hate to see that we completely stopped growing our endemic species. But maybe they need to be planted in areas where the pathogen's not. So maybe down Dunedin, somewhere a bit cooler.
Well, yeah, that certainly is a very good point. Oh gosh, I've realised. Sorry, people, I've not done my sharing the questions with the audience that I usually do as well. Managed to completely overlook that this week, but it does actually help me concentrate on reading the questions when I'm not doing that. So, and join in and help answer some of them myself, which I quite enjoy. So, apologies, but it's not all bad. Yeah, I mean, what do we plant? This is a massive question. For sure, and there is a lack of guidance out there around that at the moment, and that pathogen spillover stuff is really important to bear in mind. And of course, if we had some more resistant plants to offer, then it would certainly be helpful in that regard. More of a comment here, but very useful. The denser canopy may also result in slower host growth rates, influencing disease development. Yes, yes. We see this
Yeah.
with understory species in dense rainforest ecosystems in Australia. Interesting. Right. Is there any research on companion planting, i.e. planting susceptible species close to mānuka or kānaka with possible volatiles providing protection?
So Kānuka and Mānuka are obviously susceptible to Myrtle Rust as well. Mānuka, there has been records of it being impacted. Kānuka, nothing yet so far. Fingers crossed in the native forest. Currently we don't have studies to look at those companion plants together, but they are planted in a natural infection trial close to each other that Julia Sawato at Scion and Rod Beresford is working on. But I don't think they're looking at the volatiles or the way that they can support other species around them.
Now, a good question here. How much diversity does the pathogen population have? I don't know whether the research Michael Bartlett is doing at Scion will get to that. Roanne, do you know much about that?
Yeah, so that's still ongoing. So I've been collecting samples monthly from across the different regions, and Michael's still to process those and figure that out. So
Yes,
keep an
so
ear
that
out.
is, yeah, that is something that is being investigated to some extent. Sorry, I meant the disease prevalence in relation to canopy density. Well, hopefully that question has gotten satisfactorily answered. I personally have added many observations to iNaturalist. That's good, very good. From the native forest. Okay, interesting point. Somebody's saying here they are putting observations up from the native forest. We'll have
That's
to
great to
keep our
hear.
eyes peeled.
Yeah, we
Yes,
will.
keep going,
I know
keep
I
going.
have. There has been a few records, but there's not enough.
And a final comment here, which is excellent because I think it's about time to wrap it up. Broad scale artificial inoculations in the lab and more seedling screening and field trials would be very useful to detect variation in susceptibility in Lophomyrtus, yes, definitely.
Absolutely.
So,
And then looking for breeding for resistance as well. You know, we need to
yeah,
have that.
some artificial inoculations and screening certainly have been done. They weren't good. They weren't very optimistic, unfortunately. But yes, there could be more. And yeah, as we've mentioned, it's been seen in Australia and in other circumstances. There is the rare individual that does seem to show resistance in with these kinds of diseases. Sometimes if you're not looking, you never find it. All right, very good. And that is it today for today. And yes, thanks again, Roanne, for your presentation and your excellent efforts there tackling the questions. We've been extra fortunate, everybody, to have heard from Roanne today because she's about to change jobs. So therefore she's even busier than she normally is. Her stint at Scion is nearly complete. She's about to move to the Department of Conservation. We're delighted you will still be connected with many in the Myrtle Rust research community, Roanne, in your new role. And I'm sure everyone wishes you the best as a true champion of the ngahere. As I mentioned, a video of today's webinar and answers to questions will be made available on the website in the next few days. You'll get that by email as well. Time to announce the next webinar. We're going to hear from another Kiwi, which is excellent, and that will be on Wednesday, September the 15th. So a little bit more than a month this time. We're going to be hearing from Rob Beresford. He's a plant pathologist at Plant and Food Research, involved in several research areas, both within the Beyond Myrtle Rust research program and the Ngāraka Takitaki research program within the Bioheritage National Science Challenge. His talk will draw on classical plant pathology, so it's really going to touch on, I think, some of the questions that we had today and some of that interest from the audience, and be a really nice follow-up to Rowan's talk, because he's going to be looking forward. He's going to be looking to the future for myrtle rust here. His talk is titled Predicting Future Spread of Myrtle Rust in Aotearoa, New Zealand. So look out for the invite in another month or so. Ka kite, everybody. We will see you then.